Hello and welcome to an overview of mitosis and the cell cycle. So there's lots of reasons why cells undergo mitosis or cellular division. One might be during reproduction and the growth of a fetus. You start off as one cell, that's a fertilized egg and sperm coming together, and you eventually become something like 35 trillion cells. So reproduction and growth of organisms is really important, but also things like tissue renewal. Every 7 days, your epidermis or your exterior skin lining is replaced. Every month, the internal parts of your digestive system are replaced. Your bone cells are constantly being renewed and broken down as you age and grow based on calcium intake in your diets. Other things like going to the gym and lifting weights and inducing hypertrophy or muscle growth can be a reason why you might add more cells or have more cell division. And the organization of cells in terms of genetic information are chromosomes, and this is where the genes that correspond to proteins are found in the nucleus. And it's going to be really important that every time we divide a certain type of cell, whether it be a muscle cell or an epithelial cell or a cone cell in your eyes, that the two cells that arise from one have duplicated, completely identical information. That will be important for not only having adherence to making specific proteins, but also making sure that we don't have any problems arrive from genetic abnormalities. And the thing that you need to know about a chromosome is that when the chromosomes are duplicated, we have sister chromatids. So this is two copies of the same chromosome that are attached at the centromere. And as we go through mitosis, we are going to go from having one chromosome to two copies of the same chromosomes known as sister chromatids attached at the kinetochore. And then during the process of mitosis, we're going to be splitting those two sister chromatids into two discrete but identical chromosomes found in two daughter cells arising from the parent cell. Now, the cell cycle of cells generally is known as both interphase and M-phase. M-phase is going to be the steps of mitosis, finalizing in cytokinesis, and then we have G1, S, and G2 phase. G1 is just normal living conditions of a cell. S phase is where DNA is duplicated, and then G2 is getting ready for mitosis. Now, in terms of the steps of mitosis, generally we can start with G2 of interphase, and during that time, we're going to have our chromatin that's been duplicated in the nucleus, and they might start to be condensed or organized. That's going to happen by the time we enter prophase. And the other thing is that we have two centrosomes with our centrioles, and these are where we are going to be having microtubules being created, polymerized from these centrioles, and these are known as asters. Aster means star in Latin. So we're going to see over time these microtubules expand and radiate outwards almost like rays of sunshine. And when we get into prophase, we have our chromosomes now organized into sister chromatids. So we have discrete organized package of genetic information. You can see the centromeres really easily at the sort of middle point of the chromosomes. We have our asters radiating out with our mitotic spindle. So again, these are our microtubules that are being polymerized. And by the time we get to prometaphase, we've had somewhat of the initial dance occur. So we have our microtubules attaching to the centromeres, specifically at regions called kinetochores. And these are proteins that allow the polymerized microtubules to attach. We have non-kinetochore microtubules, which are going to be sort of like they're going to be used to expand and push away the two pairs of sister chromatids from each other. So they're going to be expanding the cell and helping with cell division. And we'll also notice that the nuclear envelope has been fragmented, so it is kaput. So we get to metaphase, and metaphase is going to be the fourth step and the most important step in mitosis, because in order for the cell to be forming two identical copies, we need to make sure that all of the sister chromatid have been duplicated and copied. So we can see that we have our centrosomes at the poles of the cell. They have microtubules attached to each pair of sister chromatids at the kinetochore, and we sort of have this equatorial plate on or within the cell where all of the duplicated genetic information is. Cells will not go further until they have signals that tell the cell that they are good with all of these being lined up on the plane. Now, anaphase is going to be where we have separation of the sister chromatid. So the important thing to note is that we're not removing or deleting any genetic information, but because the sister chromatids have now been divided, we have two copies of the chromosomes forming two daughter cells. So here we have our chromosomes that are sister chromatid, two copies within one chromosome. Now we have two copies of identical information that have been divided. Important to note is that as anaphase goes into telophase, the non-kinetochore microtubules, these very long strands of protein, are going to be pushing against each other, helping sort of push the two poles of the cell apart, while at the same time the kinetochore microtubules are going to be depolymerized, sort of pulling in the sister chromatid almost like a fish on a reel. We get to cytokinesis, which is going to be where we've formally separated the two pairs of genetic information. The asters have sort of been depolymerized, so we don't see any more kinetochore microtubules being formed. And the nucleus and the nucleolus is being formed because cells are going to start making proteins to sort of fill in the new house that they moved into. So this is just another image showing how the microtubules attach to the chromosomes from the centrosome. And again, they're being attached at the kinetochore region in the middle of our sister chromatids. And when we see our kinetochore microtubules being depolymerized, experiments have shown that the depolymerization occurs at the kinetochore as a motor protein moves down the microtubule. So while it's easy to think that the reel is being shortened at the centrosome, it's actually at the kinetochore where the microtubules are depolymerized. This is what a cleavage furrow of a cell looks like, and this is due to a thin band of actin and microfilaments, so proteins sort of squeezing the two sides of the cell almost like a belt until they undergo cytokinesis. Within plant cells, because of the rigid cell wall in plant cells, rather they have vesicles that form a cell plate that will become a new part of the cell wall, and they do that after they have divided genetic information appropriately, and that will then form two new plant cells as that new cell wall forms. So cells in plants are going to undergo mitosis first, and then they're going to have the cell plate form as a result of protein expression. And this is what we can see in microscopic slides of plant cells as that cell plate is produced. Now, in terms of bacteria, because they're prokaryotic with a lot less genetic information, their mitotic experience is different in the sense that they have one singular circular strand of DNA that is going to be made into two copies, and that generally occurs at what's known as the origin of replication, where we're going to be copying this entire strand of DNA starting at one specific spot. As that replication continues from that initial spot, we're going to see that the two copies of the genetic information is going to be pushed apart, which helps create the space and volume for cytokinesis to occur. And that's going to result in two daughter cells. This is a lot less complicated. It's a lot more temporally efficient in terms of occurring on a much faster scale, but it's also a lot more error prone, which is why sometimes bacteria can evolve far faster, because as bacteria do this, they're copying down genetic information very fast, and they don't necessarily have the same signals and checkpoints to make sure that all of the genetic information is copied, because they are doing genetic duplication and cell division at the same time. Alternatively, when we get to eukaryotic cells, the S phase, or the genetic information duplication, occurs separately from the cellular division phase, and G2 is full of checkpoints to make sure that the S phase has been completed with fidelity and good genetic copying skills. Generally, cells will have certain signals within their G1 part of their life cycle, which will either tell them to go ahead with cellular division, hey, we need some new skin cells, or they will not receive information, which means they become sort of cellularly arrested in G0, meaning they're not going to undergo cell division. And this is due to concentrations of cyclin, which is a cell signal that can be used to target cells that need to undergo cell signaling. So we can see that generally the cyclin concentration builds and builds and builds until we reach M phase, and then it drops substantially during the G and the S, and then it starts to build up during G2. And this is going to be based off of a receptor CDK, which is in cells, that is allowed to bind to the cyclin, forming a association called MPF. That's going to be degraded, and then it's going to be recycled. So generally, the only way we get a cell cycle completion is by accumulation of the cyclin in more and more amounts. Which leads us to cancer. So cancer is when cells undergo uncontrolled rapid cellular division, and this is generally due to the fact that there isn't a complete set of checkpoints that are being adhered to, or their cells aren't being able to communicate with cell signals properly. And that generally leads to the development of a tumor, which is an uncontrolled region of growth within cells that don't have the normal checkpoints. A benign tumor can metastasize and become malignant, which means that it can form tissues in other parts of the body as the cells divide and grow and spread through blood vessels. And it's really important that different treatments are applied to people with cancer early on to prevent the growth and spread of these cancerous cells, because once they start growing, they can exhaust nutrients and tissue supplies and other aspects of healthy bodies without having any checks or balances. I hope this has been informative for you to understand the applications and the processes of mitosis. Let me know if you have any questions. Thanks.